Good day and welcome again to yet another very entertaining video. As we continue our dive into the realm of reproduction, today we want to focus on female reproduction, and we're going to break this down into a couple of different things. We want to talk about folliculogenesis, oogenesis, and the uterine cycle, and how they're all related, but how independently different they are and what each term means. And today's topic is folliculogenesis. And of course, the wording simply means development of the follicle. We're not going to focus on development of the oocyte or the egg, but rather all the structures surrounding the egg. Now, where do we get started? As a common background, I'd like to start where we've started most of our endocrinology studies, and that's in the brain with the hypothalamus. Here we've got the hypothalamus, and it's releasing neuroendocrine molecules, hormones, into the hypophyseal portal system. Right, if you recall, we've got a portal system that consists of two capillary beds in series, and the hormones entering the portal system, in this case, we want to talk about a hormone called GnRH. It stands for gonadotropin releasing hormone. GnRH will travel through the portal system, exit the next capillary bed, and is now in a prime position to stimulate the production and release of two hormones from the anterior pituitary. Let's draw our anterior pituitary. Of course, the posterior pituitary on the back end is just really an extension of the hypothalamus. But here in the anterior pituitary, this is glandular epithelial. And under the influence of GnRH, we get out two hormones that will enter the blood and travel through the body, LH and FSH. Now, if you watched the previous video on male reproduction, you'll recognize that these are the same hormones, and it's for the most part the same signaling axis to get here. We'll definitely show some gender differences, but the hormones themselves, LH and FSH, are doing very, very similar things. The target for LH in the female is a cell called the thecal cell. And analogous to the Leydig cell that we saw in male reproduction, the thecal cell has the responsibility of producing androgen. This androgen primarily comes as a form of testosterone called androstenedione. FSH targets a different cell type. The cell is called the granulosa cell, and this would be analogous to the Leydig cell that we saw in the male counterpart, and in many ways serves the same purpose. The granulosa cell is there to help facilitate the maturation and protect the oocyte during its maturation until the oocyte is ready for ovulation. Now, we do see some differences in how the system is regulated, certainly in males versus females. If you recall, in males, testosterone production by Sertoli cells, by Leydig cells, was the key regulator providing inhibition on GnRH and LH. Similarly, we see that feedback inhibition from androgen onto LH and to some extent onto GnRH. But because we're talking about androstenedione instead of testosterone, this is much more weak, only about 12% of the total potency we would expect to see with testosterone. So we'll call this weakly inhibitory. And what that means for us is it's not that big of a deal. There are other key regulators that we want to focus on. Perhaps the most important hormone that we want to talk about today is estrogen. Now, where does estrogen come from? Well, if we follow the androstenedione, the androgen produced by the thecal cell, this hormone is going to be taken up by granulosa cells. And here we see a significant role for the granulosa in regulating this system. Under the regulation of FSH, and with the help of the androstenedione produced by the thecal cell, the granulosa cells are going to convert the androstenedione into estrogen. So one of the things that the granulosa cells do is increase estrogen secretion. And it's important to recognize that they're not producing estrogen from scratch, but rather they're converting the androgen from the thecal cell into estrogen, and this conversion is done by an enzyme called aromatase. So they have increased aromatase activity. Estrogen has a really important job to play here in the feedback regulation of GnRH. But I can't tell you that it's positive or negative because it's both. So it could be inhibitory, and it could also be stimulatory. In other words, estrogen has a biphasic influence on GnRH. It's inhibitory primarily when estrogen is low, but at very, very high levels of estrogen, it becomes stimulatory, and we'll show you how that works and why it's so important in just a few minutes. Because of this biphasic response, we actually have a really interesting positive feedback mechanism built, where FSH, acting on granulosa cells, causes increased estrogen production, which then feeds back to regulate GnRH, sometimes causing increased levels of FSH in order to build estrogen secretion. Now, we still want to uncouple everything that's going on with FSH and estrogen from the LH and the thecal cell and everything on the other side. And so we still have a hormone called inhibin, whose job it is to specifically target FSH. And while all this stuff is going on, what else does FSH do? Well, we're going to see increased proliferation. And importantly, we're going to see an increase in LH receptors. This is important because initially granulosa cells are not responsive to LH. Remember, LH targets the thecal cells. But during this positive feedback mechanism, FSH is building LH receptors so that at the right time, it will also become responsive to LH. So let's connect this, but with an asterisk to remind ourselves that usually granulosa cells are not responsive to LH, but there is a key point at which they become responsive, and that's going to be the LH surge, which we'll talk about in a minute. All right, now that we've got the signaling, for the most part, diagrammed out, let's go back in time years to about 6 weeks post-fertilization. In this tiny little female embryo, we see the migration of a cell type called a primordial germ cell, a PGC. This cell migrates in from the yolk sac and populates an area called the gonadal ridge, near where the kidneys are going to be developing, and immediately secretes surrounding mesenchymal cells to come surround it. This process of covering is called investment by what we call somatic support cells. These somatic support cells are later going to become the follicular cells that we're going to be talking about. But I want to start here because at this point, the primordial germ cell, it'll undergo a few rounds of division, but then it starts the process of meiosis. And again, we're still looking at about just a few months post-fertilization of this embryo. That primordial germ cell represents the next generation. In other words, this embryo's children. And that primordial germ cell will start meiosis and it will arrest in prophase 1. And now we call that cell a primary oocyte. It remains completely surrounded by a single layer of squamous epithelial cells, these somatic support cells, and the primary oocyte goes dormant. And we'll talk about what happens to this oocyte in our next video, but this oocyte is going to remain a primary oocyte all the way up until the LH surge. In other words, the egg itself doesn't change for years and years and years. It's going to remain dormant. What we want to focus on is what happens to these other cells, these follicular cells. Because at this stage, what we have is called a primordial follicle. It's interesting, at 5 months development, right? This is prior to birth. A female embryo has roughly 7 million primordial follicles. Over the next 4 months, many of these are lost, so that at birth, the female is born with about 1 million primordial follicles. Assuming mechanisms of natural selection that we don't truly understand, there's a significant die-off of many of these follicles. And then throughout the early stages of life, there's a continual drop-off in the number of primordial follicles, so that at the beginning of puberty, the average young woman holds about 400,000 primordial follicles. And that's where we like to pick up the story. How do I get from this primordial follicle to a mature Graffian follicle that will ovulate and present an egg for fertilization? Now, if we zoom in on this, let's make sure we understand where we're at. This is all happening within the ovary. This diagram shows an ovary and highlights the various stages of folliculogenesis that we want to discuss. We have the primordial follicle we start with here. And what I really want you to again appreciate is the fact that in all of these pictures, the oocyte inside remains dormant. It remains a primary oocyte even through ovulation. Now, at the onset of puberty, every month, between 30 and 50 primordial follicles are chosen to begin this journey of maturation. Only one will make it to the end, Again, there's this idea of natural selection, some sort of process that's only selecting the healthiest, the most vibrant of these follicles. But the entire process, from primordial follicle to mature Graffian follicle, is going to take approximately 5 months. Now let's highlight some of the key things that we want to mention. By the time we get to a primary follicle, there's a couple of things that are important to note. We've seen increased proliferation of these follicular cells, but now we're going to call them granulosa cells, and we introduced this cell type at the beginning. And you also see the addition of this fuzzy little yellow line now surrounding the oocyte itself. This is called the zona pellucida. If you're eating M&Ms right now, think of it as the hard candy shell. This is proteins from the oocyte, polysaccharides, sugars from the granulosa cells. This is going to be an important part at the time of fertilization and regulating polyspermy and all sorts of other things. So the zona pellucida is an important structure. As we next transition over to the secondary follicle, you can see now multiple layers of granulosa cells, but you also see some connective tissue that contains a new cell type. We have on the outside a structure called the theca externa, and on the inside a theca interna, and these contain a new cell type, the other cell type that we introduced at the beginning, the thecal cell. It's still unknown actually where these cells come from. It could be that they're mesenchymal cells, fibroblasts and such that are redifferentiated to become thecal cells, or that it represents a dormant thecal population that is recruited from the ovary at this time. We don't fully understand. But again, just to remind you, these are the cells that respond to LH to produce the androgen that's going to be used to make a lot of estrogen. Now finally, let's look at our mature Graffian follicle. We can see in here this fluid-filled space right in the middle that we call the antrum. This is important because we've separated linkages of these granulosa cells in preparation for ovulation. The remaining cells here that still surround the oocyte, we call the cumulus oophorus. Think of cumulus clouds. This is a nice soft cloudy covering surrounding the oocyte. And the oocyte actually won't lose this and hatch until after fertilization takes place. And it's this mature Graffian follicle. Now, remember, this entire process from primordial follicle all the way to mature Graffian follicle, and we've only highlighted the key changes. There's lots of different subsets that we haven't mentioned. This entire process takes about 5 months. And so on a monthly basis, again, 30 to 50 of these primordial follicles are recruited, and then during the next 5 months, they're slowly weeded out, and several of them, 1 to 3, maybe 4 of these could reach Graffian follicle stage, and then one Graffian follicle is chosen for the ovarian cycle. And so at the time of ovulation, there's just a couple of things that I want you to see. The oocyte is still surrounded by the cumulus oophorus. There's still that primary oocyte in the middle, arrested, but it's about to be woken up. It's been like that for years and years and years. And the other thing that I want to highlight is this structure right here. The remnants of the ruptured follicle become the corpus luteum, which is the main source of production for now primarily progesterone, but also estrogen. This is the main sex hormone secreting gland in the female body at this time. And in our next video, we'll discuss how the fimbriae of the fallopian tube will scrape that primary oocyte surrounded by the cumulus oophorus right off the surface of the ovary and pull it into the antrum of the fallopian tube, which is the site where fertilization is most likely to take place.